Hello viewers, my name is Sumaya Iqbal. Welcome to this tutorial on missense variant to protein structure analysis web suite, shortly MISCAST, that we developed at Broad Institute of MIT and Harvard. MISCAST is a platform to visualize and analyze missense variants on protein structure. Further, through MISCAST, user can explore protein functional, physicochemical, and structural features that are perturbed by the amino acid substitution and may have a biologically meaningful impact on protein structure and function. We have two different tracks in MISCAST, variant summary reports and variant analysis suites. The tracks are accessible from the home page through these buttons and from every page through this drop-down menu in the top navigation panel. The main navigation panel also has a documentation tab and an About tab. In the documentation tab, we gave the link to the YouTube playlist where we will be subsequently adding tutorials, resources, and showcases of MISCAST. So stay tuned. After that, we described the protein features that we used to characterize the pathogenic and population missense variants. We mined and annotated the amino acids with 7 different features, having subtypes adding up to 40 features. User can scroll down and read more about the features in this page. In the About page, we have a bit details about MISCAST and its data set, and the description of 2 primary tracks. After that, we gave the contact and citation information. Our paper will be out in bioRxiv soon, and we will be announcing the DOI after it is available in bioRxiv. Then we have the data usage and disclaimer policy and an acknowledgement note to the development team. So let's go inside the variant analysis suite. From this page, you have 2 types of selection, by gene name and by protein class names. We have 1,330 genes listed here under the gene list and 24 protein classes listed under the protein class list. So I will go for the gene selection first. I'm selecting P10. In the gene-wise page, we have 6 different tabs. Information tab shows up general information about P10, such as uniprot the name of the protein, protein length, and then the count of missense variants. Now, the count is given for the full gene and also the number of variants that were mappable on 3D structure. We provided these counts for population variants collected from NOMAD database, which is the genome aggregation database, and for pathogenic variants collected from CleanVar database and Human Gene Mutation Database, or HGMD. Next, we have the protein class information for P10. All genes have been categorized in all classes and then in their respective functional classes. P10 has been annotated as in all classes as hydrolase and phosphatase. After that, we provided the features that were found to be associated to pathogenic variants and population variants. Respectively, for the functional classes, P10 is annotated with all protein classes, hydrolase, and phosphatase. If the users are curious about the statistical analysis, we refer you to go through the paper listed under the citation info of About page. In the one-dimensional page, we have the linear protein sequence annotated with variants. Starting from the top of the page, we have shown some general data such as protein info, the transcript for variant information, and the protein class information. Then you see the linear amino acid sequence. When you slide this gray bar, you can see the full sequence. The color code here is for variant types. Blue indicates there's a reported NOMAD variant in this position. Red indicates there's a reported CleanVar or HGMD variants for this position. And orange indicates there's an overlap of pathogenic and population variants on that amino acid. To see the annotation for an amino acid, You can either select, click on the index, or can double click on the amino acid. So after you open up the amino acid specific information, three blocks of data for that specific amino acid pops up. The first block of data gives you the variant information for that amino acid. If there's a reported nomad variance, likewise for valine 45, we show the substitution and the allele count, allele number, allele frequency information. Now, if we have a position where there's a reported clean var or AGMT variance, then we show the substitution along with the phenotype information. In the next block, you can see the protein features of tyrosine 68. Such as tyrosine 68 forms a beta sheet in 3D structure. It has zero solvent accessibility surface area, so it resides in the protein core. It's an aromatic amino acid. There's no protein-protein interaction annotation available for this amino acid. Then the spatial distance from this selected amino acid to the known post-translational modification site. And we show this information for different protein structure available in protein data bank for the selected gene. So I will explain one line here. So this line shows in the protein structure 1D5R chain A, on the structure position 125, there's a known acetylation site, and the distance to that acetylation site from the selected amino acid tyrosine 68 is 11.9 angstrom. So in our study, we assumed if an amino acid is within 10 angstrom distance to a known PTM site, that substitution can be functionally meaningful. However, if the substituted amino acid is itself a known PTM site, we specifically mention it. For an example, Y68 is a phosphorylation site, so the distance is 0. Finally, the functional feature annotation information from UniProt, where we can see that tyrosine 68 is part of the phosphatase tensine modular domain. In the next block of information, we give the comparison of the features of tyrosine 68 with protein class specific pathogenic and population variant associated features. Such comparison is shown differently for each functional class information. Here I will explain one table. The first column shows the feature category. The second one shows the features of tyrosine 68. And the third and fourth columns show pathogenic variant associated features and population variant associated features. Now, a feature of the selected amino acid is highlighted in red if it is a pathogenic variant associated feature, like here. And the feature of the amino acid is highlighted in blue if it is found to be associated with population variants for that specific protein class. Such details, we believe, will be helpful to hypothesize the pathogenicity of the substitution of this amino acid. Next is two-dimensional visualization tab, where we show two-dimensional plots with variant and feature information. And we have separate plots for 7 different features here. In each of the plot, the x-axis shows the amino acid index and the y-axis shows the feature subtypes. For example, for 3-class secondary structure plot, the y-axis labels are helices, beta strand or sheet, and coil, and there is another label for the amino acids where there is no structure available. The variant types are color-coded in the same way: blue for population variants, red for pathogenic variants, and orange for both types of variants. Now you can mouse over to the plots to see the specific amino acid and index. You can also do some other stuff from this toolbar here, such as you can zoom in and you can zoom out. You can do the selection of a specific part. You can click on this home button to get your access back, and you can take a snap from this camera icon. Likewise, the plot for 8-class secondary structure. For accessible surface area, in this plot, the y-axis shows the numerical value of the accessible surface area. Now, in this plot, here we can see that there is a good cluster of pathogenic variants here at the core of the protein. The other plot for which the y-axis is the numerical value is the distance to the post-translational modification types. For an example, you can see here that we have many amino acids where we have different types of variants and that are within 10 angstrom distance to known post-translational modification site, which is here specifically a phosphorylation site. And these amino acids here are the known phosphorylation sites where we have also observed pathogenic variants. Next comes the plot for functional features, where we can see that we have a pathogenic variance in P10 that is right on an active site. And we have a couple of both types of variants in an annotated region and modular domain. Next, 3-dimensional visualization tab. In this page, we have simultaneously showed the features and the variants in the linear space on the left side and on the 3D space on the right side. The linear viewer is separated by Plot Vista wizard, and the 3D structure is shown through Lightmall viewer. From the drop-down menu here, you can select your protein structure of interest. You can see if this structure is a single chain structure or a complex structure, and you can highlight the variation position, such as these are the C-alpha carbon position of the amino acids where we have pathogenic variants, and you can turn it off. You can simultaneously select 2 different types of variants and compare their spatial distribution. From the 1D space, you can also select a specific feature type to highlight that feature type on the structure. So on the 3D viewer side, we have some other exciting features here, like you can zoom in, and then you can open the control bar to do some in-depth analysis online. And importantly, you can download the protein structure with all the along with all the annotations of variant position and features to do your offline analysis. And when you open the downloaded file, you will see this view on your PyMol space where you have the variants and all the 7 features that I described is available in MISCAST are all saved as separated objects. In the feature table tab, user can see variants and features in tabular view. Importantly, user can download the annotation of variants and features per amino acid as a plain text file where the variants and features are available in a tab delimited fashion. For better visualization, we have shown only the 4 columns by default, but you can select additional columns here and to view it online. For an example, here we see 2 pathogenic variants available on this amino acid. If we want to see the detail, we can bring in this column here to see specific phenotypes. And also we can go to subsequent rows by selecting different pages from the bottom of the table. In the index table page, we have given the genomic index to protein sequence to structure index mapping. In the genomic index, we have given the transcript information, chromosome number, and the 3 codon position. And in the structure index, we have given the protein data bank ID, chain identifier, and the position of the amino acid on structure. So with that, we finished the tour to gene-wise page of variant analysis suite. And now I will take you to the protein class page of the analysis suite. So I'm selecting the protein class kinase here. In the information page of protein class, you can see the definition of the class, the genes that are annotated as kinase, and then the population missense variants and pathogenic missense variants counts of kinase class that were mapped on the 3D structure. Next, you see the pathogenic variant associated features and the population variant associated features for kinase. In the visualization tab here, user will find the statistic and analysis output for this class on different features. For an example, for 3-class secondary structure, on the left side, you see a bar plot which shows you the proportion of population variants and pathogenic variants on coils, beta strand or sheet, and helices. On the right side, we have the forest plot showing the output of Fisher's exact test, computing burden of different variant types on different features. Now, if the odds shown with the circle here is greater than 1, then we have an enrichment for pathogenic variants on these specific features shown in red. Otherwise, the enrichment is observed for population variants, which is shown in blue. Important to note that such association is significant when the p-value is shown with an asterisk or the confidence interval bar is black. Otherwise, when the association is not significant, then the confidence interval bar is gray and there is no asterisk sign. When the feature value is numeric, then to show the data statistic, we have used distribution plot. So here you see the distribution of accessible surface area for pathogenic variants and population variants, and that distribution is statistically significantly different. So pathogenic variants stay much more in the core of the protein with less accessible surface area. We have now finished navigating through the analysis suite, and I will take you to the variant summary reports. In this page, first we need to select the gene name. I'm going for P10 again. And then some general data pops up. After that, we have to select the amino acid index. With that, the report for that specific amino acid pops up. So first, we gave some variation information that I have already explained. So I'll directly start explaining the reports. So in the second block, we gave the quantitative report, and then comes the detailed text report. So let's see the quantitative report. So in MISCAST, we evaluated the burden of pathogenic and population variants in a total of 40 protein features. Now, with those identified features, what we can do is we can compare the features of that selected amino acid here with the identified pathogenic and population variant associated features and can give a quantitative score. So let's read out one report. For phosphatases in general, we identified 7 features that are significantly associated with pathogenic variants and 8 features that are significantly associated with population variants. Tyrosine 68 of P10 possesses 4 out of 7 pathogenic variant associated features and 0 out of 8 population variant associated features, giving MISCAST feature difference spectrum value of 4. Now, whenever this value is positive, this indicates that the selected amino acid possesses higher number of pathogenic variant associated features, and when it is negative, then it has higher number of population variant associated feature. Such a score is visualized also through the box plot below. After that, we have the detailed text report. And in this detailed text report, you can read that what are those protein features that were matched with the significantly associated features to gain biological insights into the pathogenicity of the variants or to hypothesize the molecular level impact of the substitution. So with that, we have finished taking the tour to MISCAST. We will be uploading more videos on how to use the 3D viewer and application showcases, so stay tuned. I hope you like our web suite. Thank you for the patience.